Welcome everyone to this D-BSSE eSymposium. It's my great pleasure to welcome you here. Today we will dive into the world of machine learning in medicine. The topic or the title of our symposium is Predicting Clinical Phenotypes with Machine Learning. I'm Carsten Borgward. I'm the professor for machine learning at the Department for Biosystem Science and Engineering of ETH Zurich. Our department is located in Basel. This symposium today is part of a larger series of eSymposia that my department is hosting. Today it's my turn, but we encourage you to stay informed and to stay connected. You can receive information on the next D-BSSE symposium by sending an email to the email address shown here with the subject eSymposium. You can also subscribe to the D-BSSE monthly newsletter. You can follow us on Twitter or on LinkedIn. The program today consists of 3 talks. I will give an introduction to the topic of clinical phenotype prediction with machine learning. And then we have 2 exciting external keynote speakers. Patrick Schwab from GlaxoSmithKline will present, and Mikhail Rosensweigh from IBM Research. We are very much looking forward to these talks. I'll tell you about the speakers and their topics in a few minutes. So the first talk is this introduction. The second talk is the one by Patrick, real-time prediction of COVID-19 related mortality using electronic health records. And then Mikhail will talk about drug repurposing from prediction to causal inference, identifying unknown benefits of existing drugs from retrospective patient data. So with this, we'll go right into the midst of things. I will present our work and the topic of predicting clinical complications. Let me just switch the slides for that. Here we go. So what is this field of predicting clinical phenotypes with machine learning all about? To understand that, I would like to take one step back and discuss, first of all, what we mean by machine learning in this context and what we mean by medicine, and in particular personalized medicine in this context. Machine learning is the search for statistical dependencies in large data sets. So it's a combination of applied statistics on the one hand, the search for statistical dependencies, and of computer science, so how to efficiently handle large data sets. And the health domain provides an enormous wealth of data for this purpose. But what are the kind of statistical dependencies that we are interested here in this domain? Well, it's the statistical dependencies that are the basis of personalized medicine. We try to find statistical dependencies between patients' properties and patient phenotypes. And we try to use these for improved diagnosis, prognosis, and therapy decisions. We want to tailor medical treatment to the properties of each patient by exploring the statistical dependencies between often molecular patient data and higher clinical phenotypes of a patient. Now, if you look at this research topic of machine learning in medicine, then 3 big themes, 3 big clusters of topics stand out. The first big group or cluster of topics is the automation of diagnoses and treatment. And a very good example for that is this study from December 2017. It's an automatic system for recognizing metastases on a slide. So it's the detection of lymph node metastases in women with breast cancer. And it's an image recognition task. So you get a slide that represents a sample from these lymph nodes. And now the task is, traditionally for a human expert, to count the metastases on this slide. And doctors get better at this task the more experienced they are. Often after years of experience, they become very good at this task. And in this paper, the authors presented and developed an automatic, a computer-based approach to this task, so a computer vision algorithm for this task. And it turned out that already in 2017, this automatic computational system could recognize these metastases as well as new doctors with a few years of experience, and even better than very experienced doctors, so with almost a decade of experience, if these doctors were put under time pressure. The only group of doctors that could outperform the computational system were those experienced doctors that were not put under any time pressure. And this system has been further developed. So I communicated with the first author yesterday. So companies have been founded around this topic that really try to bring this computational system now to the market. So that's one prime example for automating with machine learning a task that was previously done by human doctors. And the same holds for treatment. There are attempts to find models how to treat complications in the intensive care unit by learning from records of how human doctors treat the same patients. Another big topic is the topic of biomarker discovery. The big distinction between the second group and the first group is that in the first group, we are not necessarily creating new biomedical knowledge. We are rather trying to mimic what a human doctor can already do. This is different in this second big cluster. So in biomarker discovery, we are really striving to detect new statistical dependencies, new links between patient properties and patient phenotypes. And the example shown here is a paper that will be presented today by Patrick Schwab in his talk. It's the real-time prediction of COVID-19 related mortality using electronic health records. So predicting whether someone might die from COVID-19 based on the electronic health records. And this is a new kind of biomedical knowledge. So as you know, COVID-19 was a new disease last year. So we didn't know which factors influenced mortality. Here, machine learning contributed to building a predictive model for the risk of mortality in COVID-19 patients. Another form of creating new biomedical knowledge is to do drug repurposing, to find new uses of existing approved drugs. And that's what Mikhail Rosensweigh in her second talk today will talk about. Another example of using machine learning to create new medical knowledge from existing data. A third big branch of this field of machine learning in medicine is biomedical data management. And I mean this in the broadest possible sense, from genome assembly to managing electronic health records at a national scale, or even beyond that. Here, there are machine learning problems in every step of the task, because we are dealing with such enormous data volumes. There's a necessity to automate this data management wherever possible. And one example that also illustrates the big economic and commercial importance of that is this acquisition of Flatiron Health by Roche 3 years ago. So Roche paid $1.9 billion to acquire Flatiron Health, which is a company that is dedicated to providing cancer patient treatment data in a structured form. So the founders realized that very often cancer treatment data is not stored in a structured fashion, and the mission of that company is to change that, to create harmonized ways of how we store information about cancer patient treatment, and that after a few years of existence of this company was worth almost $2 billion to Roche. This gives you an idea of how big the commercial impact also is of this field of machine learning in medicine. So now what I'm going to describe in the remainder of my introduction today is one topic within this second field of biomarker discovery that is very dear to my heart and to my research group, namely the early prediction of sepsis. Sepsis represents a global health crisis. 27 to 30 million people per year develop sepsis. It's a major cause of death in intensive care units. 7 to 9 million of these people die. So that means every 3.5 seconds, one person in the world dies from sepsis. And even survivors of sepsis may face lifelong consequences. So we are interested in finding biomarkers for sepsis in order to be able to predict sepsis early. But why is there a need for biomarkers for sepsis? Why is it of clinical relevance to predict sepsis early? Well, let's first define what sepsis really is, and then it will become apparent why early recognition of sepsis is so important. Sepsis, according to the international consensus definition sepsis 3, is a life-threatening organ dysfunction caused by a dysregulated host response to infection. And now the relevance of early recognition stems from the fact that bacterial species identification in blood still takes between 24 and 48 hours. So I should add, sepsis, this infection, is often treatable with antibiotics, but identifying the bacterial species in the blood that is causing the infection is still a rather slow process. So it takes between 24 to 48 hours. Each hour of delayed and effective antibiotic treatment increases mortality in sepsis patients. Therefore, recognizing sepsis early allows to treat sepsis early and thereby increases the survival chances of the patient and reduces the damage to the patient. Detecting and treating sepsis earlier is therefore of highest clinical interest. There's a famous statement by Machiavelli from Il Principe, which in fact also applies to sepsis. It's about hectic fever, so fever with a high variation. And Machiavelli said hectic fever at its inception is difficult to recognize, but easy to treat. Left unattended, it becomes easy to recognize and difficult to treat. And exactly the same holds for sepsis, and that's exactly the reason why we are developing machine learning methods for predicting sepsis early. Now let's describe the problem a bit more formally, or algorithmically at least. What is the input of our machine learning algorithm? What is the input? Now, the input is the rich data that is being collected about each patient in intensive care units. So that can be lab tests, that can be the health history of that patient, but in particular, it is vital signs, physiological measurements such as body temperature, heart rate, blood pressure, respiratory rate, oxygen saturation, and other physiological measurements that are constantly being monitored in an intensive care unit. So we have long, real-time, high-dimensional time series that describe the state of a patient. And now, given these observations, our goal is to predict sepsis-related phenotypes. When will sepsis start? Will sepsis start? And when will it start? So onset of sepsis prediction. Will there be a particularly severe form of sepsis, a so-called septic shock? Or will the patient even die from sepsis? These would be three typical examples of phenotypes that we try to predict using machine learning. Now in my lab, we have taken two perspectives on this problem. The first one is an association mapping type of approach. So here we are interested in finding motifs, in finding patterns in these time series that are associated with developing sepsis. So if you're familiar with genetics, you know the term genome-wide association study. We are developing techniques here that are in analogy to what is being done in genetics on SNPs, so on genotypic profiles. We are trying to find similar patterns that are outcome associated in time series and in high dimensional time series. And we have worked a lot on this topic over recent years. The other perspective is maybe the more classic one, and that's the one I will focus on today, namely to predict sepsis in time series using classification approaches. And this is the dominant and primary approach in the field. However, as you will see in a minute, it's a very complicated task, and there are many pitfalls that you encounter when you try to do sepsis prediction using time series classification. And I'll discuss and describe these now in the following. So even a few years back, when we started our work on sepsis prediction using machine learning around 2018, there was already work out there trying to do the same using classification algorithms. So given the data for a patient, will that patient develop sepsis, yes or no? That's the classification problem one tries to solve here. And at the time, there were 4 main papers out there, and they differed in many characteristics from each other. So they were they used different data sets, as you can see here. They used different definitions for what constitutes sepsis. So like the old consensus definition, sepsis 2, but not exactly, but a related definition. One study used the new consensus definition. 2 other studies used the ICD-9 codes, so retrospective annotations of the time series that this patient had sepsis, and then an ad hoc criterion to determine when that patient had sepsis. The different approaches taken varied between deep learning models to more data mining based representations of changes in these time series. This is this so-called InSight algorithm. And 3 hours before sepsis onset, these different methods reported different areas under the ROC curve in distinguishing patients that develop sepsis from patients that will not develop sepsis. So this reached from 76% area under the ROC curve to 96% area under the ROC curve. And the prevalence varied greatly between these data sets. So in the rarest case, 6.6% of all patients developed sepsis. In the Futoma et al. Work, 21.4% of the patients developed sepsis. So varying prevalence is another aspect that differs between these different studies. There was also already one randomized clinical trial to use one of these techniques in order to check whether using such a machine learning-based early warning system for sepsis would increase patient survival and reduce the hospital length of stay. But what we noticed along the way when analyzing these existing publications was that there's great heterogeneity in the definition of sepsis and how it is implemented. There was even a study that showed that various sepsis definitions lead to different cohorts. So it's not minor changes. It's not that we go from 2,000 sepsis cases to 1,900 sepsis cases. If we go from one definition to another, sometimes this changes by an order of magnitude. So it's not minor changes. One problem I already alluded to, it's the fact that several authors use ICD 9 billing codes, a sepsis label. This is a general label for the time series without exact time of sepsis onset information. And then they use an ad hoc criterion, like for instance, signs of inflammation in these time series to define the sepsis onset. And even more critically, even if people use the same definition, sepsis 3, on the same data set, we observed that the number of sepsis cases varied greatly between different studies. So with the same definition and the same data set, we observed 1 order of magnitude difference in the number of sepsis cases between different studies. Now, how can that be? How at all can that be? The reason is that there are various ways how you can implement the sepsis 3 definition. The sepsis 3 definition includes 2 factors, 2 criteria that must be met. There must be a so-called suspicion of infection by the doctor. The doctor must suspect that there is an infection. This is either done by antibiotic treatment or by body fluid sampling from the patient. And then in a time window around the suspicion of infection, there must be an increase in a so-called SOFA score. This is the sepsis-related organ failure assessment score, so a score that quantifies organ damage. There must be an increase in that. If we have both of that, the suspected infection and the increase in SOFA score, then we fulfill the sepsis 3 definition. But the devil is in the details. So what is the baseline, for example, for this increase in the SOFA score by 2 points? What is the baseline that you start from when quantifying this 2-point increase? Is it a whole baseline window, as we did in one of our studies? So is it a baseline window 2 to 5 days prior to the suspicion of infection? Is it the value 2 days prior to the infection? So defining this baseline value is one reason why there is heterogeneity in the implementations of sepsis 3. And then when both criteria are met, which of the 2 defines the onset? Is it the increase in the SOFA score, or is it the time point of the suspicion of infection, that is the actual onset of sepsis? So there's a lot of lack of details here that can result in different implementations of this sepsis 3 phenotype, and therefore lead to these different number of cases that can even differ by an order of magnitude. So to summarize this part, there are enormous challenges when it comes to predicting sepsis using time series classification. It's extremely hard to compare different studies to each other. They vary in how they define the labels or how they define sepsis in this case. They vary in how they extract the label, so how they implement the label definition on a given data set. Therefore, the number of cases can even differ on the same data for the same sepsis definition. Reproducibility is a major issue. It's not only the case that often the prediction algorithm is not available, it's also the case that the code for the label extraction is not available, so you cannot even check how exactly this label extraction or this implementation of sepsis 3 has been done. There's always a danger of circularity in these tasks. As you have seen, some of the temporal observations that we make, like the increase in the SOFA score or the suspicion of infection, are used to define the sepsis onset. We must be careful not to use the same observations for predicting sepsis onset. So we always have to cleanly separate these two. The measurements, the observations used for predicting sepsis, those for being used for defining sepsis onset. Another big question is how to define the controls. So what is a control patient here? Is this a sepsis case before the person gets sepsis? Is this another person that didn't get sepsis at all? When exactly do you then set the pseudo onset of sepsis in this control patient? So you need some reference point also within these controls to be able to tell whether you are able to successfully predict sepsis in advance. So these are major problems in the evaluation, the definition of the controls. You can make the task much harder or much easier by different definitions of controls. And few studies report precision recall values despite considerable class imbalance between sepsis cases and controls. We've written a systematic review on all of these challenges, which just came out yesterday. So Frontiers in Medicine, Moore et al. You can read up on the details of all of these points that I've explained here. Now we tried to develop our own model for sepsis prediction that stays as close to the original sepsis-free definition as possible. And we imputed missing values in the time series using multitask Gaussian processes. And then we used the deep learning approach, temporal convolutional networks on these time series to predict sepsis. And we compared this to a more classic data mining approach, namely dynamic time warping using k-nearest neighbor. And we did this on Mimic3, so one big public data set for these tasks. We had 570 sepsis cases and 5,618 controls then in our study. And we observed the following. So if we look at the right here, we see on the x-axis hours before sepsis onset, between 7 hours before sepsis onset and 0 hours before sepsis onset. And with the best method, we compared here 4 different methods to each other, 3 deep learning techniques and this one data mining technique. With the best method, we reached more than 85% area under the ROC curve, 7 hours before sepsis onset. So 7 hours before sepsis onset, we can with more than 85% probability tell a person that will develop sepsis in the next 7 hours from a person that will not develop sepsis in the next 7 hours. And also in terms of area under the precision recall curve, we reached very good values here, so 0.4 for the best method. Now, interesting to us back then was that the best method was not one of the 3 deep learning approaches. It was the classic data mining method that reached here the highest area under the ROC curve and the highest area under the precision recall curve, 7 hours before sepsis onset. Only in terms of area under the precision recall curve, if we move closer to the onset of sepsis, the 2 of the deep learning approaches get on average better than this data mining approach based on dynamic time warping. But it is a reminder to not always take for granted that deep learning will be the only possible and best solution to a problem. Deep learning is extremely powerful, but it's also important always to compare to a classic baseline. So to summarize the key points of my talk, when we do complication prediction using machine learning, what I said holds for sepsis, but for many other medical complications as well, then there are inherent challenges regarding comparability, reproducibility, circularity, and proper evaluation. There's an enormous challenge when it comes to imputing missing values. So missing values are very common in these time series. Therefore, working on methods that can directly deal with irregularly sampled time series are very promising. It's one research direction that we are working on. Deep learning architecture matters. So we saw differences between the different deep learning architectures that are being used for this task. And we were reminded that using classic baselines is a must-have in such studies. you should never miss to have a classic baseline. Now, in our next steps, we are trying to develop these methods at a national scale. So together with Adrian Ekli from the University Hospital Basel, I'm leading the Personalized Swiss Sepsis Study, a consortium of all 5 university hospitals in Switzerland, the corresponding universities and ETH, in which we try to develop algorithms for predicting sepsis and sepsis-related mortality. There's a phase 1 in which we are using electronic health records for this task. This is heavily ongoing, so we've now collected more than 10,000 electronic health records all over Switzerland. And there's a phase 2 that we are now starting, in which we try to do the same using omics profiles on hundreds of patients. And in a single-site collaboration with colleagues from ETH Computer Science around Gunnar Rätsch and colleagues from Inselspital Bern around Tobias Merz, we have developed a similar system for predicting circulatory failure. And this turned out to be very successful. So we can discover 90% of all circulatory failure events with 1 out of 3 alarms being correct. So only 2 out of 3 alarms are false in that case. And 82% of these events we can even detect at least 2 hours in advance, such that we leave time for the practitioner, for the clinician to intervene. This is shown here. So we have a circulatory failure event here shaded in red. We have a period before that event during which we would like to warn the clinician of an imminent event. So we define this to be 8 hours here. So if the alarm goes off within these 8 hours, that would be a correct alarm, a true alarm. If the alarm goes off before that 8-hour window, that would be a false alarm. And if no alarm goes off during this 8-hour window, then this would be a missing alarm. So then this would be a missed event of circulatory failure. If there is an alarm, it is a captured event. And then in this system, we determine precision as the ratio of true alarms over all alarms. And we determine recall as the number of captured events over all events. Good, so this is like a single site pilot study for the things that we also want to do in sepsis next. So to conclude, automation, biomarker discovery, and biomedical data management will remain key research topics in machine learning in medicine. We'll learn about 2 exciting examples of biomarker discovery in the next 2 talks. What we'll face in the future is an enormous data growth in 3 dimensions. So there will be population scale data sets of individuals. We are moving towards lifelong recordings of health state, maybe not in a few years, but over the next few decades, we are moving towards this point, and we are moving to a point at which we have highest resolution information on the health state of an individual, down from the molecular level up to the higher phenotypic level. The big question for machine learning and medicine is how to mine, how to handle and use this data. And many branches of the life sciences face very similar or analogous problems. So it's clear that machine learning and medicine remains a very exciting field. I thank my team, our collaborators, and our sponsors. And as you are interested in this topic, I would like to point you to the Marie Curie Initial Training Network that we are coordinating on machine learning frontiers in precision medicine, and its homepage, mlfpm.eu. So we are hosting online symposia and also online summer schools here in this as part of this network. And if you want to listen to talks about this topic, not only for a few hours as today, but for a whole week, then you are most welcome to join these events. Thank you very much for your attention. And now it's time for questions. So I'll stop my share screen. Here we go. I'll go through the questions one by one. I hope you can hear me. So I'll read out the comment by Stefan Basser. Thanks for the nice talk. Did you try to predict the bacterial species causing the sepsis, since different species can require different antibiotic treatment interventions? Well, not in this project, not in Moore et al., but we have an ongoing collaboration with the University Hospital where we try to do exactly that. But it's not published yet. But we are exactly interested in this topic. Could you please elaborate a little on the imputation method you mentioned in your summary slide? So we used the multitask Gaussian process for this task. So we used the Gaussian process to impute missing values in these time series, and we treated the different channels, the different variables as related tasks. You mentioned the choice of controls, control onsets in sepsis prediction, and that you can make the task much easier or harder. Can you elaborate on that? What would be hard, easy choices? Yeah, so one choice that would make the task much easier is the following. If you take as controls patients that did not develop sepsis, and then you define a pseudo onset point in these patients, which is close to being discharged from the ICU. This is not so unlikely because, well, as you know, there must be this time horizon before the event during which we want to make predictions. So this pseudo onset time in controls cannot be right at the beginning of the stay in the ICU. So there is a risk that the pseudo onset time that we set in the controls is towards the end of the stay, is towards the time of being discharged, and then the algorithm will not pick up that person will not develop sepsis. It will rather pick up that the person developing sepsis is in a bad health state and the person being discharged is in a good health state. And this is actually a confounder we see in many of these medical complication prediction tasks, that the machine learning algorithm is often able to pick up generally getting better or generally getting worse. And you must be careful not to turn the sepsis prediction problem into a getting discharged from the ICU problem, because that is much easier. Okay. Can you potentially apply this time series analysis also on long-term physiology data, for instance, as collected by sport watches, to predict unknown conditions or risk factors for a given patient? Absolutely. This is absolutely something that one could do with these algorithms, or for instance, with the association mapping algorithms. I know about studies where sleep patterns around the globe are being analyzed with such smart watches, and it's exactly a form of time series mining that is happening there. Good. So then there are some more questions in the chat. I'll now move to these. When you say it's a very high dimensional data set, can you give us some idea of how high dimensional it is? Thank you very much. Yes, so it's not a microarray level high dimensional, but it's dozens or hundreds of features that we are dealing with here. So in Moore et al., depending on what we looked at, it was above 40 or around 70 features that we looked at. So in the order of 10 to the 2 to 10 to the 2. Good. And are there shapelets indicative of sepsis onset? Can this be extended to other disease time causes as some form of disease shapelet atlas? Yeah, that's actually something we would like to create. So maybe I jump to the first question first. So are there shapelets indicative of sepsis onset? Yes, so we have one publication, this Bock et al., where 2018, where we discover a few statistically significant shapelets in sepsis patients, but only on one data set yet. So we have to validate these findings in more data sets, but we discovered statistically enriched shapelets there in people who develop sepsis. Can this be extended to other disease time courses such as some form of disease-shaped atlas? Yes, so that would be one like goal of mine, to do something like this, to collect an atlas of disease-associated shapelets. That's exactly one of the purposes of this line of work. Good. So with this, there's one more, and this is then the last one from Lucas Meander. Thank you for such a great talk. Is algorithmic fairness typically evaluated in published sepsis predictors? Is data approximately balanced between, for example, different ethnicities? So it's not, so in most of the papers we have been looking at it, this algorithmic fairness has not been evaluated, but Thomas Gumsch and I recently commented on using these clinical severity scores, so like a predecessor of these prediction systems that we are currently developing in different ethnicities. So there was a paper, in fact, by a co-author of Patrick Schwab, saying that these scores work differently well and so with different levels of accuracy in different ethnicities, and that they might underestimate or overestimate the risk in certain ethnic groups. So so we commented on this work in Lancet Digital Health. This is a very important topic to consider in the future. Good. So with this, I thank you very much for your attention and this very interesting round of questions. It was very stimulating. And now it's time to move on to our first keynote speaker of the day, to Patrick Schwab, who did his PhD at ETH Zurich. So he's an alumnus of our school. He then joined Roche and Genentech and was a machine learning architect there before very recently moving to GlaxoSmithKline, where he's now the director for machine learning in medicine. So very fitting, and he developed this very exciting real-time predictor of COVID-19 related mortality using electronic health records. Patrick, we are very happy to have you here, and we are very excited to learn more about this work of yours.
Awesome. Thank you so much, Carsten. And I could not have asked for a better introduction. And thank you so much for setting perfectly the scene here for my presentation. I'm very happy to be able to present to you today our work on real-time prediction of COVID-19 related mortality using electronic health records. This is work that we did at the start of this year and has actually been published very recently in Nature Communications. I would like to acknowledge our great team here, incredibly talented team, cross-disciplinary, hailing from the Max Planck Institute in Tübingen, Harvard University, Beth Israel Diakoness, the Kantonsspital Winterthur and Kantonsspital Uniklinikum Tübingen, and also my colleagues here across the world, essentially. And acknowledging also here our funding partners, Roche, SNF, of course, and NIH, and Cypher as well. And the topic that we're really addressing here is one that you are all too familiar with at this point, but the situation is still on a global scale quite massive. So we're looking at something like 170 million COVID-19 cases here, and already a count of around 4 million deaths globally stemming from the COVID-19 pandemic, which is placing a significant burden on healthcare systems worldwide. And what this has led to fairly directly, which has been fairly stark here, for example, here in the US case, in terms of the actual utilization of intensive care unit and healthcare resources in general across here the US context, but we're seeing the same phenomenon spreading across the globe, also towards emerging nations. India, for example, is a very recent example where we're seeing very, very high utilization rates in excess of 70%, for which there's actually good scientific evidence that this leads to worse care overall being provided to patients. And so the problem that we're really looking at here is once a COVID-19 patient is diagnosed as having COVID-19, there's many different scenarios in how they can be treated. And there's different levels in terms of the severity of their disease, in terms of what level of care they would receive. And if you look at the levels that they could receive potentially, There's the major group of the outpatients, which would be the patients that receive some sort of care in the hospital, but then are not ultimately admitted. And this is really the majority of patients, actually, so roughly 70% looking at US data longitudinally. We also have people that actually get some form of admission into a hospital bed. In a hospital, actually, it is roughly 5%. And then lastly, the last group here is the group that actually gets admitted to critical care because their severity of disease is really, really high. And so this also corresponds roughly, and this is what we would like to see, roughly corresponding with the mortality risk that those patients would be likely to experience. And for outpatients, this is actually a little bit hard to estimate because obviously the records there are not as good as for those patients that have been actually admitted to hospitals, but it's likely a substantial amount of people that actually have mortality events outside of the hospital. And there's some evidence of research coming out recently around a 3 to 4-fold increase of out of hospital sudden deaths during this pandemic period, which is a substantial absolute number. And we're also seeing some biases towards socioeconomic groups and also minorities in these numbers. So this is really a group that we should focus on and where we could potentially do more for patients. The second group here really is the admitted group, which has roughly a mortality rate of around 5.5% overall of all of the people that get ultimately admitted to hospitals. And lastly, the group that is admitted to critical care units, there the mortality rate is really excessively high, something around like 25% or more. And really the problem that we're trying to solve here is how do we prioritize these levels of care to the right people at the right time? Ideally, we would not like to have any people dying at home, for example, without receiving the right level of care at hospitals. This is a problem because we also have limited resources in the sense that we cannot actually treat everyone with the highest degree of urgency. And these high utilization rates already tell us we are actually utilizing the entire capability of the healthcare system in many contexts. And so really the question we ask ourselves in this project is, can we utilize real-world patient data at longitudinal scale to better predict individual COVID-19 related mortality risk in order to make that assessment, in order to help people assign the right level of care to the right patient at the right time? And the rough overview of what we do is really summarized in this one slide here, where we start out with data about a COVID-19 patient. So these are all patients that have been diagnosed at some point with a laboratory test. And then we feed that data into an assessment model that essentially gives us a prediction in terms of what the likelihood of that patient's mortality experience in the near term is. We then visualize and analyze this data in order to make inferences about these particular patient groups. And in terms of the input data, we look at electronic health records. Electronic health records, for those of you that are not familiar with this data, are extremely rich electronic representations essentially about the clinical pathway that a particular patient has been taking. In particular, these data points include clinical observations, diagnoses oftentimes, and also lab tests and vital signs and biosignal monitoring data that is really collected at longitudinal scale here for these patients. We've been lucky to have access actually to a very, very large sets of data covering roughly 70,000 COVID-19 diagnosed patients in roughly 70 healthcare institutions in multiple continents and countries, covering the US, Malaysia, India, and Australia. And this is really fairly representative and one of the largest data sets that have been evaluated in this context to date. The model that we use is actually a neural cox survival model that essentially leverages these high-dimensional representations of the patient's status, clinical observations, demographics, diagnosis, etc., in order to make that survival prediction, and is updated actually in real time every time a new data point about that patient comes in. There's a reassessment in terms of what that risk now looks like for that particular patient. I will be going into a little bit more detail about this in the next few slides. And the last thing we do is actually we visualize and analyze this data, present it to the decision makers in order to actually make something out of this decision in terms of assigning the right level of care, increasing the urgency of care, etc., for these patients. And I will be going into detail exactly how this looks like in the next few slides. And this is really one example here that I would like to go a little bit more deeply into. This is actually a high-risk patient that has been assigned a very, very high risk in terms of mortality by this prediction system. And so this is really the output that you see here from the system. And at the top, you see several time series that we take, a selected set actually. So this is not the entire set, but a selected subset of these signals that are fed as an input. And essentially, these time series give you an indication of how this patient is developing along different dimensions of their clinical status. You can see actually on top of each of these time series, a colored bar. This colored bar actually assigns a degree of importance to these different input sources in terms of how important and how much do they contribute to that final score of the prediction system. So they can give us some idea in terms of what the decision of the model is ultimately based upon. And if we see a red area here atop of these clinical time series, this would correspond to a negative increase, so an increase in the risk of that particular patient, whereas a blue contribution would mean this has contributed positively to the score in the sense that the risk of the patient because of the score has decreased. And what is quite interesting here is if you look at, for example, the SpO2, so this is blood oxygenation measurement that is taken for these patients at a high resolution, we can actually see here that before the intubation event, this is actually a negative contributor. So this is increasing the low oxygenation rate in the blood here of that patient is increasing actually the contribution of that prediction to the risk. Whereas after that intubation, key intubation event happening around 150 hours here from onset of disease, you can actually see that it becomes a positive contribution because the O2 level has essentially stabilized for that particular patient. Short time before the death event, actually, that happens here, roughly around the 300-hour mark. Actually, we see this again becoming a negative contributor because it dips below a significant level here for the oxygenation in the blood of that particular patient. We also see key inflammatory markers, white blood cell count, creatinine, that are actually increasing and giving a negative contribution over time as well, especially prior to this death event that we observed here around this 300-hour mark. And at the bottom here, you can actually see the entire magic of the system working, where essentially you see this COVID early warning score plotted here every time a data point updates in this electronic health record of this particular patient. It starts out with roughly a 60 percentile compared to the entire cohort, which is already a fairly high level even from the outset for this particular patient. And you can see actually this rises over the course of their stay, even towards this intubation event. And actually this intubation event does not overall decrease the mortality risk of this patient. And we see actually that this risk is something around the 90th percentile. So this is extremely, extremely high level for this particular patient. And what is also interesting here is we see that the system actually predicts this very high 90 percentile score in terms of the risk for that particular patient, roughly a week before that ultimate mortality event that actually happens a week later. And so we can actually see that there is an opportunity for potentially intervening earlier by having this information available at an earlier time point. What's also interesting here is you see this time series atop of this COVID-19 score, which is a Sulfur early warning score, which is a general purpose mortality prediction score that is used within intensive care units as a kind of clinical baseline that is not COVID-19 specific. And you can see actually here the advantages very well of the COVID-19 early warning system, which is very much tuned to the COVID-19 condition and takes into account a much larger set of factors in real time, essentially. So it reacts even to minute changes in this patient status and reflects them immediately in this scoring system here. I would also like to highlight a second example here. This is actually an improving patient, in some way a contrasting example to this previous example that we've seen. And so this patient here actually comes in similar to the other patient with this roughly 60 percentile. And so from the outside, they're quite similar in terms of their status, but we see actually that this patient's markers improve over time. So we look at, for example, inflammatory markers like the C-reactive protein here, which initially towards this 200-hour mark within the stay actually is a negative contributor, but over time stabilizes quite quickly and becomes a positive contributor throughout the time frame. And we also see the respiratory rate and everything related to the oxygenation stabilizing as well and becoming more robust over time. And so all of these factors are taken into account by this COVID-19 early warning system at the bottom again. And you can see actually a steady decrease in the risk of that patient over the course of the stay, roughly around this 200-hour mark, where potentially this patient was, I would say, out of the woods in terms of their mortality risk. So this is a roughly 5th percentile score, which is a fairly low risk. And so this patient potentially 9 days before their actual discharge could have potentially been discharged a little bit earlier, so could have freed up resources for another patient that might have needed these resources a little bit more. And so this is exactly these two cases that I wanted to highlight where we have misallocation potentially of resources. So we have patients that could receive a higher degree of care, and we also have patients on the other end of the scale where potentially we could have assigned a lower degree of care based on their high resolution risk scoring here that we see, for example, coming from this COVID-19 early warning system. Here really an opportunity for reprioritization, I would say. The system was evaluated in this sort of pipeline, and I don't want to go into too much detail here, not to bore you, but essentially we're looking at both an internal validation using a held out test set, but also an external validation using a completely independent set of data coming from other hospitals, other countries in some cases, in order to really test whether or not the system is generalizable to new clinical contexts, which is really a potential bottleneck and a potential problem in a wide-scale deployment of these systems. And so we really try to evaluate this across a wide range of potential scenarios. And one plot that I wanted to definitely show you here is whether or not one question that we really wanted to answer is whether or not the system is really able to, on a fine rate level, assign the right level of risk to the right patient group. And this is a survival curve stratified by the COVID-19 early warning system score here. And you can see actually in deeper blue colors, the patients that have been assigned a higher risk percentile, and the deepest blue corresponding to this 90 to 100 percentile risk score that you have seen, for example, for this very high risk score patient that I've shown initially as a qualitative example. Whereas these patients that have a lower risk here in the lighter blue color, and you can see actually this is the 0th to the 60th percentile in this case. And you can see actually this is a fairly well distinguished plot in terms of the personalized risk for these different groups. You can see actually that the risk is markedly increasing as we go towards higher percentiles. And this is in the test holdout test set cohort that we look at. And here, particularly, the patients that have a very high risk do have a markedly higher mortality and should potentially be treated differently than patients that have a lower score. We also looked at the prediction performance in terms of the specificity at actually finding those patients that ultimately have a mortality event using a sensitivity of 95%. So really trying to capture the majority of all patients that have mortality events and how many of these are false alarms essentially. And you would ideally you would have want to have 100% real true alarms in terms of the system versus detecting the right mortality events here. And we see actually here these two plots here correspond to the two cohorts that we evaluated. On the left hand, it would be the internal held out test set that we looked at. And on the right side would be the external validation that we performed on different hospitals and different patient cohorts from different countries in some cases even. And we can see actually here in deep blue, the COVID-19 early warning system compared to several non-COVID-19 specific systems, but also some recently developed COVID-19 specific systems that also in some cases leverage machine learning. We can find that COVID-19 early warning system is actually markedly higher performance in terms of predicting which patients will receive ultimately mortality events in those cohorts. And this seems to also generalize across both the internal validation, but also the external validation here on the right side with other patients from other clinical environments. The other thing that we evaluated is actually a split in time, in the sense that we finalized our model at a given time point, and we then collected more data in the future, and we checked that whether or not these temporal dynamics, i.e. whether or not the change in how we treat patients and how we learn about treating COVID-19 patients in general, but also how the pandemic develops, variants develop, etc. And so we looked at, does our system maintain its performance over time, taking into account all of these different effects and that happen over time. And so we finalized the model and we looked at over a period of something like 4 months, is our model's performance actually robust given all of those changing dynamics? And here you can actually see the performance of that in that particular cohort in the future, in some sense, where we can actually see that the performance was well maintained in terms of the predictive performance for identifying those mortality events, also well compared to state-of-the-art models in different domains. The other group that I wanted to highlight here is the group of patients that was ultimately not hospitalized. So they were diagnosed, but they did not receive hospital care. So potentially the group that we could do a lot for in terms of reprioritizing, so avoiding out of hospital mortality events essentially. And here you can see actually the performance is also fairly strong. And so this points to us towards believing that a system like this could potentially be helpful in actually identifying those patients and potentially assigning hospital beds to them because they might be receiving mortality events out of hospital if they were not receiving that admission at some point. We also looked at, and this goes to a question that I heard in the Q&A session beforehand, we also evaluated, because we had such a representative data set across many different ethnicities, We also evaluated different backgrounds and minorities and socioeconomic backgrounds and everything within this paper. And we actually looked at, is the performance of our model robust to these different subgroups? And this has been a problem that has been highlighted in literature quite heavily and recently in terms of the model's potential biases towards these different cohorts. And we wanted to make sure that the model, if it is deployed, it would be robust to these particular subgroups, and it would have the same, maintain the same level of performance for many different, as representative as possible for the people that actually ultimately receive COVID-19 diagnoses. And actually, we found that in this setting here, we found relatively robust performance across all of the different subgroups that were present in this particular data set that we had access to. We also looked at what are actually the driving factors for a particular patient's risk to be higher and heightened in this case for mortality. And we identified actually a very large set. And so these are all the features that we looked at in general. And we also find that there's a fairly long tail in terms of important features for predicting. And if we take, for example, here a p-value cut off of around 5%, we find that there's still a set of roughly like 20% predictors that are relevant, statistically significantly relevant for predicting COVID-19 mortality. That's not to say that these other variables don't add predictive performance, but they might not reach statistically significant on the entire cohort level here for this particular data set. But we see factors such as age, well-known factors such as age, SPO2 obviously, and biosignals related to inflammatory markers, as we've seen, for example, in those qualitative examples previously, have been fairly highly scored by these models. But also interesting factors such as prior diagnosis, kidney diseases, for example, intubation is a big one, creatinine levels, hypertension, etc., being very high predictors, and also LDH score here from the lab values, a high predictor. We also see that the long tail, and this was surprising to me, is fairly long for this disease. So we see a large set of factors actually that for some patients have a high predictive value, but don't reach that significance level for the entire cohort. So I think this really goes to show why it's important to go beyond a small set of features and really look at a comprehensive set so that we can actually make the best decisions for the largest amount of people. And this is really a key takeaway that I would say we had from this study in terms of what takeaway in terms of guidelines for designing these systems could be. I would also like to briefly touch upon the clinical translation and integration of these systems into actual clinical workflows. And there's really two ways this could be done in terms of taking a system like this and implementing it in actual clinical care. And the two main ways I would like to outline today is really the EHR integration. So actually taking a system, an algorithm like this, and putting it directly into the electronic health record system and displaying it alongside information that is already present in those systems to clinicians potentially. And then a more lightweight app solution, for example, where it could be a standalone application that would not need as much deep integration here with the hospital side. And really, what could this look like in terms of the EHR integration here on a very high level? Essentially, there would be some 3 stages that would be have to be part of the hospital infrastructure here. And so the first step really is about data ingestion, data standardization, where essentially you have to make sure that you receive as a model standardized and clean data in terms of an input level features that are used essentially as the foundation, as the basis of the predictions of this particular mortality prediction model here. And then we really need to have in place in the hospital infrastructure the right set of compute and a model deployed essentially on that hardware in the hospital. And then lastly, there would have to be some integration in terms of the visualization and the display of that information to clinicians. And you can see that on the left here on this slide, particularly where, for example, it would highlight here not only the score, but also the temporal dynamics of that score and how the different time series and clinical markers actually influence that particular score over time. And really what is still missing, and this is also highlighted in this slide here, what we have not yet done is actually a prospective validation in the sense that actually trying to connect if the system was available to clinicians, is this actually accretive to better outcomes? So having the system, does it improve finally the patient's outcomes, which is ultimately the goal that we have with the system? And does it bring better value than caring for these patients without this particular system? The second really main approach that you could conceivably follow for integrating a system like this into clinical care would be a more standalone application. And here's really one main difference to the previous model that I described, where essentially you would replace that automated integration within the electronic health record with a more manual data input This has the advantage of not requiring that integration, which as some of you might be familiar with, is actually a fairly laborious step that is not easy to facilitate. Whereas here you could actually have a manual data entry. The negative side of this particular solution would be that entering this data is obviously quite burdensome for clinical staff. And especially if you want to evaluate it at different time points, at multiple time points, it might become quickly a time limited factor. And so you might actually prefer, in terms of convenience, the integrated solution. But in terms of implementation, it is much easier, obviously, to have the standalone solution that potentially can be called on demand instead of in real time in this particular electronic health record system. I would also highlight a little bit the limitations around these studies. In particular, this is a retrospective study. And so we did not actually apply the system and then look at what the performance of the system is. Rather, we took the data from a large period of time, covering a relatively representative set of patients, and retrospectively developed a prediction model that is able to identify those patients in that data set. So it's not clear yet whether or not employing that system actually in that cohort would yield better results and would yield better outcome for these patients. And so we would ideally have to test this post facto and actually test this in clinical settings to be actually sure that this helps and that it's efficacious in terms of improving clinical outcomes. The second really big limitation of the study is that there are often limited options. While this is improving over time now, there's often limited options for actually intervening and acting upon these information points. So even if I knew that the risk of a particular patient's is heightened, I might not be able to act upon this. So I might not actually have a treatment available that really would help change the course of that particular patient. So this is always a bit of a downside. If I don't have a better way of treating those patients, even knowing that they have a heightened risk, might not help me manage that patient better. And so the third big risk here is really also one that is associated with all mortality prediction tasks. Essentially, many patients in the ICU particularly die not due to the disease underlying, but rather due to cessation of treatment. So we decide that it's perhaps time to initiate end-of-life care for a particular set of patients because it's not worth anymore keeping them alive just through machines and treatments, etc. And so this is really a pitfall of mortality prediction. One could argue that many of these predictions are actually made in terms of predicting patients for which the cessation of treatment would happen. And so this is actually a confounding factor that we're aware of in this particular study. And the last really big limitation of the study is that we do not have good death records for patients that die in the outpatient setting. And so if they are not tracked within the hospital, obviously it's harder to get those mortality data points. And so this is a big limitation of these particular studies as well. So to summarize, we really developed this COVID-19 early warning system using data from more than 66,000 patients from 70 healthcare institutions across multiple continents and across multiple countries as well. Using the electronic health record, it accurately predicts individual COVID-19 related mortality risk in real time and could just potentially improve care prioritization for those particular patients. There's many follow-up opportunities in terms of prospective validation, clinical translation, research into treatments and outcomes, and actually correlating this with this particular score. So there's really many potential avenues for further research in this topic, and it's becoming more and more timely, obviously, as the pandemic continues and spreads across the globe. And so I would like to break here and thank you for your attention. And I would be happy to take any questions you might have regarding this topic.
Thank you, Patrick, for this exciting talk. A lot of questions have been asked in the question and answers window and in the chat. I'll go through them. I'll read them out, and we can answer them interactively.
Thank you.
So by an anonymous question here, thank you for the great talk. Where did you obtain your data from? Is it public or accessible upon signing agreements? Also, was the code published?
Mm-hmm. Great questions. First question around the data. This was a fairly large longitudinal cohort coming from 2 federated networks of hospitals. So these are data aggregators essentially that work with the hospitals as partners to aggregate on a national scale, in some cases, data sets regarding certain patient groups. This was the particular cohort of the so-called Optum research data set. And then the second cohort was the TriNetX research data set. That both are these types of network aggregators. They both are not, as far as I know, public data sets. And so you need to have a data sharing agreement essentially with these partners to get access to this. And it might involve some licensing fees as well in some cases. So this is unfortunately not public data, but as far as I'm aware, some of these data sets is actually available as part of the big national cohorts of COVID-19 research data. So you would have to look into this, but I think they also contribute to these data sets. And so I think ultimately you might be able to get access through the public domain in some ways, but if you directly contract them, I think you would need a separate agreement essentially for access to this data. So great question. The second one around the code is publicly available on GitHub actually. GitHub link is within the paper, and we have made public this entire code for actually developing this model and running this model. And we hope to essentially make it as accessible as possible. Obviously, as pandemic advances, we would like to have this tool as widely available as possible.
And if I may follow up on that, Patrick, so is the predictor, the trained model available, or is it that the scripts are available for training the model if you had the data?
Mm-hmm. Great question. The model itself is not public simply because we could not make the data itself public, and there are some concerns in terms of deriving potentially some of these data points from a trained model. So it's not publicly available. I might be able to make it available on request to particular research interests. So I can definitely be receptive to any requests there, but it's not generally available in terms of a download that you could just get, but data in terms of the model itself and the code to deriving it, if you have a data set to run it on, are available to general use.
Good, thank you. Then next question by Caroline Weiss. Slide 19, how did you include the outpatients in the prediction when they are missing continuous clinical monitoring and EHRs, heart rate, temperature, etc.?
Mm-hmm. This is a really, really great question. So 2 points to this question. One point I already mentioned is these data are much more sparse, and we also have in the paper, you will find all the details in terms of how the missingness patterns change for these particular patients. But essentially what we looked at is in a realistic scenario, so given only the data points that we have for those patients, so this is really trying to work with as much data as we have for each of these patients at that scenario. And so they might have, for example, presented at hospital at some point for getting their laboratory tests, for example. And so within those steps, there might have been a few data points collected, but generally much less data, much more sparse than you would find, for example, for patients that ultimately get admitted, or even patients that get admitted to intensive care units. So you will find that these patients have actually much less data. And so you can actually see this encouraging performance in terms of what you see here in this graph here is actually reflective of the missingness. So this is the performance given that missingness, which is even more encouraging because it shows that even the information that we have available for those patients, which is much less than we have for this intensive care unit and admitted patients, it is still enough to actually make better predictions in terms of identifying who might ultimately have mortality events. This is all obviously under the caveat that we don't have comprehensive records for those patients. So it might actually be that some of those patients for which we don't have a mortality flag in our data set, they ultimately ended up still having a mortality event that was not recorded because we did not have the death record. And so there's mixed levels of quality in terms of following up and trying to find those death records after the fact if they have not been observed in the hospital. So this is an entire issue with these observation cohorts where essentially it's a missing data problem where we don't have all of the death records. And so this is under this caveat, but essentially for those patients where we have some death records, it seems to be able to still identify them, which is encouraging to some sense. But obviously, we would like to have some prospective data in terms of being able to actually validate this in a prospective cohort where we have clean data in terms of we know that we have captured every single death event.
Thank you, Patrick. Another anonymous question. You said a prospective clinical study is still missing. Is it planned for other diseases than COVID then, I assume?
Mm-hmm. This is definitely something that we are still in conversations with. One of the problems here is obviously to roll this out, you would have to do this translational steps, which especially the Ehr integration is fairly intensive in terms of the work required and IT infrastructure and everything. And so it has been a little bit difficult actually finding partners that are willing to invest at that particular time to actually developing and deploying this and then running this clinical study. And so we haven't actually yet started this particular prospective evaluation, but it would be something that we would be actually very, very eager to running. And we would ideally want to do so in multiple contexts. in general, I think the second part to this question in other diseases is also, I think, a really, really interesting direction, where essentially you can see these types of models. So going beyond just COVID-19, I think what Carsten also correctly outlined is this is a whole research direction in the sense that there's potentially thousands of use cases where a similar principle in terms of risk prediction could be used not only for better care, but also for helping clinical staff make the right decisions, assign the right resources, intervening at the right time point, potentially earlier than they would normally do, right? So there's a lot of potential, really, really interesting use cases, I would say, where we could apply similar technologies based on similar fundamentals to different use cases. So I'm actually fairly excited about expanding the set of use cases and trying to find more and more ways we can apply these different technologies in order to improve, hopefully, healthcare, and making healthcare also a little bit more personalized and a little bit more high resolution, and really trying to leverage all of these really, really interesting data points that we have available nowadays, which really require also some kind of machine learning model to create a bridge between high resolution, high quality sensor data that oftentimes is so voluminous that actually humans cannot really make sense of this anymore in real time, where we need these bridges in terms of the machine learning models to actually make that accessible and make it amenable to clinical decision making. So I'm really excited about this actual direction, and really great question. I think that certainly points towards a very interesting future development.
Thank you. Another question by Damian Rocairo. Thank you for your talk. At the risk of sounding too technical, can you give us additional insights into how was risk estimated while dealing at the same time with missingness in the data? For instance, missing values for some covariates in the electronic health records.
So all of the technical details, I don't want to bore the audience too much, but all of the technical details are in the paper. Regarding the missingness, we essentially used multiple chain imputation to try to impute for those patients where not all values were available, essentially trying to impute the other values based on what we had available. So essentially trying to turn this a little bit around in terms of trying to predict what was missing for these particular patients. And because we had fairly representative data set, fairly large scale data set, we also got relatively good imputation performance for a lot of values in that data set. The second question you had was around the mortality and estimating risk. Again, the quality of the data obviously varies between the patient groups. As I mentioned, there's the not hospitalized group, which is a little bit sketchy in the sense that obviously we know that the recall there is not 100%, but for patients that have been cared within a hospital, there's actually a duty of those hospitals to record death events correctly. So I'm fairly confident in the quality of those particular data points. And that's also something that these networks of data providers work on improving the quality of. So I'm fairly confident about that level of quality. Obviously, it's a little bit more sketchy on the not hospitalized group. And so I think the risk estimates are probably trustworthy for those groups that are hospitalized. And it's not clear yet how trustworthy those outpatient mortalities are, but it's a great consideration.
Thank you. There's another question by Pekka T. It's probably a bit related to one earlier one. How were covariates collected for the non-admitted patients? I suppose the number of covariates and data collection frequency was lower than for them than for admitted patients. So,
yes, I think this is a bit of a repeat from what we just
exactly.
discussed before. But yeah, essentially for this not hospitalized group, data was much more sparse. They might have been seen for the laboratory test diagnosis. So ultimately, they have had some interaction with the healthcare system. Otherwise, they would not have a healthcare record, right? So there might be a group of patients that has never been diagnosed, and that would not be included in this data set either, right? So there's certainly a group of patients that could benefit from the system that was not included in the study. And so all of the patients that we had here, they had some interaction and they had a diagnosis at least. So there might be another group of patients that never received even a diagnosis. And so hard to say about those patients, but for those patients that had a diagnosis, they had some interaction and they had some data collected about them in the course of that diagnosis, but also previously potentially. So the electronic health record covers several years of time span. So there's actually data points in terms of prior diagnosis, et cetera, that might still be available for predictions. For example, for those outpatient groups, where we could, for example, if there's a heart disease, a prior heart disease, very high predictor, a very significant predictor for this model here, could be available actually for those patients from the past, even from previous encounters with that particular patient. And so there are some data points and there are even some historical data points for patients. And in general, the set of data points is obviously much lower, but the performance in terms of the evaluation seemed to have been still very encouraging. So I was actually very surprised that even with that reduced set of covariates, we have still been able to identify at fairly high degrees of performance these particular patients that ultimately had recorded death events in that group.
Thank you. There's another question by Thomas Gumsch. Thanks for the great talk. Did you do a kind of counterfactual retrospective evaluation, comparing patients that would have been admitted to patients that would not have been admitted?
Mm-hmm. No, that's a great question. I love this question. I think general limitation of these retrospective studies is exactly this, that we don't have access to these counterfactuals. And so a particular counterfactual that I think is very, very important that we never get to evaluate in retrospective studies is the counterfactual what would have happened if that system was in place for that particular patient. So if my clinician had this information available, would they have acted differently? Would this have improved the outcome, right? If I had this information, let's go back to this qualitative example here of this patient, this high-risk patient. Had I had this information a week earlier about the potential mortality being very high for this patient, would clinicians have acted differently? Would there have been a different treatment option that might have saved this patient, right? So this would be, from a research perspective, extremely interesting and something that we would love to know. But obviously, we don't have access to this data because it never happened in that scenario. And so I think this is actually a very, very interesting point in terms of future research that we want to do in terms of prospective evaluation, obviously. But I think there's also methodological perspective here where there might be actually ways to impute counterfactual outcomes for some patients and actually try to leverage that to evaluate in a retrospective setting still whether or not we can actually find for these different scenarios still a good performance if there is different interventions. And so this goes towards the direction of data set shifts, etc. But I think this is actually a very, very nice consideration. And thank you so much for that question.
Thank you. And another one in the question and answer window, Santiago Castro-Dao. Is there anything to be said about patient data confidentiality?
Mm-hmm. Thank you so much for that question again. I think that is always a relevant, very relevant concern. One point I want to make about this particular data set is it's all anonymized data essentially. So there's no identifying information about those patients. So we could not go back and identify a particular individual from this particular data here. And so I think this is very important that also as a guideline in terms of you can actually develop these models only based on de-identified information. De-identified here means to the standards of the regulations outlined in the HIPAA standards in the US here. And I think this is fairly important actually to ensuring that we use the data to make the best decisions for the patients, but we keep their data protected in the sense that we actually only leverage the data that is de-identified. And so we cannot actually make inferences and we cannot make this data available to malicious users, for example. And I think we actually recently, with some of the co-authors of this paper, we wrote an opinion piece as well of this particular topic, where there's actually obviously a trade-off between trying to make the best decisions for a particular set of patients with the data available, but also preventing the malicious use of that data and really making sure that we have the right balance in terms of value of those models versus risk towards patient privacy, etc. And I think this is a balance as a society that we still have to find out about more as these models become more pervasive. And I think in general, I'm quite excited about the potential health impact of actually having these models in place, but it is all, I think it has to be all under the fundament of actually having the right privacy security guardrails in place such that we actually can be sure that we're only leveraging the data in a de-identified manner. We're only making it available for these good users and we're preventing all of the malicious uses. And I think this is very important societal discussion that we need to be having about these models and the data that we collect for them.
Thank you, Patrick. There were 3 questions asked during the talk. In fact, in the chat, I'll also read these out because we still have time. By Daniel Trejo Banos, are those associations derived from your neural Cox model?
I assume you're talking about this plot here. No, these are actually not. Yes, these are actually not derived from a neural Cox model. These are derived from a linear model, essentially, to get these p-values. It's not straightforward to actually derive p-values from this neural Cox model. This is one of the losses of the interpretability in some sense of this particular model in terms of statistical analysis. So this is actually a different model that was trained, a linear model that was trained on the same data for the same task. and essentially giving you the linear association. And so it might actually be what is actually more interesting in some cases to take this information into account. And this is why this question is actually very interesting, is so this is really the cohort level information. This is why I talked about the long tail, about factors being relevant for particular sets of individuals. And actually, you have to see this in the context of these plots here. And you actually have to look at the qualitative data. You will find that if you plot not only the selected set of variables, but the entire set, you will find that actually there are time points and there are patients, individual patients, for which certain covariates are very, very influential for this ultimate prediction. And so you will actually have to look at, I think, the entirety of evidence and going beyond just the cohort level statistics here, and really find that due to this long tail, you can actually find a lot of significant increase in the performance by having more factors included, even beyond that set that is only statistically significant, because there's so many different heterogeneous scenarios, right? There's so many different patient groups, there's so many different prior diagnoses, so many different scenarios. And I think one of the powers of this particular approach here, where we have really longitudinal representative data from many different scenarios, I think this covered something like 3,000 patient hours of observation time, so much more than any human could ever see. And having a machine learning system like this to have all of the data integrated and trying to make those predictions, even in the very rare case of very combinatorial settings where there is a particular individual which benefits from a particular inclusion of a particular variable, I think is one of the guidelines. And one of the takeaways that I have from this study is that the long tail in terms of the medicine and longitudinal types of settings that we see in these types of data sets is very long. And there's actually many, many different scenarios, and no patient is the same, I would say. I've looked at hundreds of these plots. I can tell you that the heterogeneity has surprised me very much. So there's not one pattern, one typical pathway for patients that you would see. In fact, it's hundreds, if not thousands of different scenarios. And so having this type of data and being able to unbiasedly and train these models essentially on these large representative cohort is really the best way to actually leveraging a kind of learning healthcare system here, I would say, where we actually have that data available and use it for better decision making and better outcomes. And I think that is a really, really beautiful question. Thank you so much.
Thank you. There's another question by Sarah Brüning on missing data. How do you treat missing data? I think this question we had before, but the second part, this is new. Wouldn't in this case missing data potentially indicate that patients were doing well, or at least considered to, whereas additional tests, additional measures such as intubation would only be performed in seriously ill patients?
Great question. Very, very good question, actually. Shows some deep insights. Essentially, this is completely correct. So there's one weakness. So the particular way we modeled this is excluding the information about the presence of a particular value. So the model that as you see it here, it doesn't know whether a value was measured. It essentially, because we imputed all of the values prior to actually running the model itself, the model itself does not have the ability to actually differentiate between patients for which data was available and those for which it was not available. And we've chosen to do this deliberately because of this problem. If you don't do this, then there might be the problem that actually the fact of just having the presence or absence of a particular test or diagnosis or whatever, essentially being in itself predictive. And this is oftentimes not what you want, because those patterns are often not robust, in the sense that if I were to employ this model, potentially these missingness patterns would not remain the same. They would not be the same also in different hospitals, etc. So we did not want to have the model depend on these particular assumptions. And so we actually built the model deliberately in a way that it cannot make use of this information. And this is a really beautiful point. I think certainly this is something that you have to be conscious about. This is a little bit counterintuitive, because actually the performance is higher if you include these particular data points. So if you include the information about absence or a presence of a particular test, typically the performance measured retrospectively is higher, but the performance is more likely to be not robust, because there's more assumptions in terms of how tests are employed, which patients get tested, etc., baked into the model that you may not want to lean on in a deployment scenario. So a bit counterintuitive, this decision, but particularly I think in terms of robustness and generalizing to new environments, I think this is the right decision in general for making, building these particular models. Great question.
Thank you, Patrick. Now we are out of time, but we thank you very much for this very exciting talk and a very long and deep discussion that we had afterwards. So that was really great. Also, thank you for everyone who asked these insightful questions, and Patrick for giving insightful answers. Thank you very much, Patrick. And now it's my pleasure to introduce the second keynote speaker of the day, Mikhail Rosensweigh. Mikhail did her PhD in computational physics at the Bar Ilan University in Israel. She then did postdocs at UC Berkeley, UC Irvine, and the Hebrew University. She joined IBM in 2005. She is now there the director and the head of healthcare informatics at IBM Research in Israel. She's a visiting professor at the Hebrew University, and she's also a member of the Israeli National Council of Digital Health and Innovation, and leading an important subcommittee there on breakthrough technologies. So we are very happy to have you here, Mikhail, and to listen to your talk on drug repurposing.
Thank you very much for the invitation. And based on the very energetic and interesting discussion, I hope that I created a talk that fits the audience. Let's see, because I'm going to start with something and I'll move to presentation mode. Do you see my slides? Is everything okay?
Yes, in edit mode, now in full screen mode. Now it's perfect.
Okay. So I want to talk about causal inference and the usage of causal inference for drug repurposing. Before that, I want to tie this talk to the current days, and Patrick just presented this fascinating work on patients' data who have COVID and their risk. And I want to share some observations of what the COVID-19, how the COVID-19 changed our world. And I'm looking at that from the perspective of enterprises. So if we think about the changes that happened following a crisis, we can go back to World War I and to Henry Ford just before the war in 1913, who introduced the automatic assembly line for the mass creation of a car. And his innovation eliminated the need of many different experts to go to the same place for changing and adding and correcting and doing things, because he has this system of moving the things automatically and enabling the the mass production of different products. And the World War I has accelerated that innovation, and it became the standard use method of the industry, of how we in the industry create products. Similarly, the crisis of the Cold War have accelerated the adaptation of the digital transformation, and in fact, the internet prototype was created within the US Department of Defense in the 60s as a result of this Cold War. So when we look at COVID-19 and we think of how it changed the industry, we see that in the industry, the needs have changed, the behavior of clients have changed, everything has changed, and also the interest in the language has changed. So it's not only technical, is also conceptual. Everybody was talking about science and clinical trials and how quick can we get a vaccine and how you measure the vaccine success. And the world is shifting towards automating, scaling, and speeding up the world of discovery, of usage of the scientific method. The scientific method itself, of course, was introduced more than 400 years ago by Sir Francis Bacon, who talked about the scientific method, about the question that is being answered with a study that leads to a hypothesis that is being tested, and the experiment is being reported, and this is a cycle that can be accelerated, but it has specific elements and steps and processes. This was adapted by the industry only recently. So you can see, and if you think about different companies that you probably know very well, an example would be Netflix, companies who are constantly experimenting with their products. So they are not making a plan and launching a product. They are creating an hypothesis based on models on what might affect the behavior of the clients, and they experiment with their products. a resource for this realization. One resource might be the book that was published in 2020 by Professor Stephen Tomke from Harvard Business School, that talks about experimentation matters, about the fact that the industry is moving much faster nowadays in the world of digital to large-scale experimentation. So companies can experiment and accelerate the way they work in many different areas. It goes across the full value chain of an enterprise. It can be relevant for the research and development to the operations and supply chain, and for sales and marketing. And we see in different companies the usage of this acceleration of the scientific method. We actually all were very appreciative of the acceleration of the scientific method in the development of vaccine. We've seen that the companies have leveraged everything that can be used in these days in order to accelerate the discovery. And my talk, now that I just gave you this context of viewing the industry through the lenses of evolution, so we can think about the way enterprises evolve, similar to what we think about human involvement. There was the world in enterprise of local production that has transitioned to mass production and evolved into digitization and in AI. And nowadays, accelerated discovery is the way the industry is working. And I want to highlight from this very broad view a specific element of acceleration, and this is the acceleration of drug discovery. And in the world of drug discovery or the development of drugs, much efforts in time and in resources is invested in early stages, in the development of some hypotheses of what a molecule can, how a molecule can react, how a person can react to a molecule using some using some animal models and other things. And from this hypothesis, starting to run different trials and experimenting with the drug, where the very first two phases are devoted to safety. And only afterwards, there's a transition into efficacy. And when we think about all of this, about this lengthy process, it seems obvious that we would like to shortcut the process by reusing existing drugs. There are hundreds of drugs that people are using and consuming today, and the drugs are approved for specific indications against the specific condition where they can provide some remedy. But it is likely that existing drugs can be of value for indications or to support us in targeting specific health condition that is not yet known. An example of such a drug would be the reuse of the Viagra that was originally developed for some blood problems and was reused for erection problems. So that's a very famous example of drug repurposing. There are, of course, other drugs that can be repurposed, some of them already known, some of them not. As of today, the main mechanisms to identify new indications for existing drugs beyond serendipity, so just finding out that this might be of value, is to look into the mechanistic models of how the drug interacts in the body and finding potentially relevant pathways and activities within the cell that are associated with conditions not yet known to be of value for this drug, and linking and predicting based on the mechanism of action how drug might be effective against a new target. What I'm going to offer and present here is how one can accelerate the development of drugs through emulation of randomized controlled trials. And based on the discussion you had on COVID in the previous session, I know that there is here familiarity with these concepts of the cause and inference. The way the team have designed the study, and I'm going to talk on a very specific study in the context of Parkinson, the team has designed the study around users of a certain drug and non-users of a certain drug. So we are going to look into a disease. So the disease would be, in this case, Parkinson. And we are going to look at specific targets associated with Parkinson's. So it might be Parkinson dementia, or it might be Parkinson mobility problems. And we are going to look at people who have Parkinson's disease who are obtaining drugs for other conditions in addition to the Parkinson's disease, because they are suffering from multiple conditions, they have comorbidities. And I'm going to propose a system that automatically sifts through all the existing drugs in the data and identify those that seems to be most beneficial for the specific targets in place. So I guess when we move into looking into patients' data, it's clear that patients' data are highly valued. And here is a quote from the New England Journal of Medicine that talks about the transformation of medicine due to the usage of data. And it's clear that the technology that we need is not all done or ready for use. And I'm going to refer to a paper by Professor Michael Jordan and Professor Tom Mitchell, who provides very interesting perspective on machine learning, where it is today and where it's heading. And they refer to causal inference as an important element within the world of machine learning that requires further studies. And indeed, what you can see here is that we had to design the study in a specific way that I will share with you, and leverage existing and expand on existing technologies in order to obtain results with respect to candidates for repurposing for against Parkinson's disease. So the way we worked and the framework and how it was designed is available in the paper from in a paper that you can see the reference down here in a paper about what we call feature X. The first step is to extract tabular data from a sequence of events that happen to patients. We had access to data that comes from EMRs, electronic medical records, as well as from claims data managed by MarketScan data from IBM. And these data sets are data sets that you can submit a request and obtain license to access the data sets. They are anonymized and they are available for studies. And we extracted from these data some specific cohorts that represents the patients. And in order to do that, we combined the patient's data with knowledge that is available in different knowledge bases in order to be very specific and concrete with respect to who are the patients of interest. And index date would be the date at which these patients obtained the drug of interest. And for each patient, we created a history. So what would be the minimal historical period in time that we request in order to be able to characterize the patient clinical status. And similarly, what would be the period of time that we expect the drug to be effective so that we can analyze the effect of the drug within that period of time. That will be the follow-up period. And once we defined the data in this way, we were able to use the technology called feature X in order to move from sequences of data into the structured tabular data. And we used a set of tools that we made available as open source. It's called the Causal Lib. And you can see here the reference where you have the description of the information of what the scripts and code that is available in Causal Lib. And in this Causal Lib system, we are leveraging, so the starting point is this tabular data that I described before, how we calculated it. And we are offering different methods to estimate balancing positivity problems and identify outcomes and measure the performance of the causal inference. So this is just a side comment with respect to what is available for you in the event that you want to learn more about our tools. These are open source code that you can make use of. The framework is described in this JAMIA Open Data. So as I said, in this specific study on the Parkinson data, We have extracted data from the observational healthcare data, the claims and EMRs. We had a user-defined cohort of patients, and I will be very specific in a minute describing how we selected the Parkinsonian patients. And then we applied this doubly robust approach using causal inference in order to estimate the effect. And I'll provide details about that. So here you see the whole framework where we moved from what you see here is an example of how the data was laid out. We had diagnosis and treatment, and information was spread across different years, and we aligned different people around the index date, which is the date of the first time that we see that a certain drug of interest was made available to the patients through prescription. And we have analyzed the data, and I'll show here how we looked at this specific Parkinson's disease patients. So the definition was patients at a certain age, because there's a big difference between young people with Parkinson's disease and elderly people. So it was people who are at the minimum 55 years old, and that they have diagnosis associated with Parkinson. We required that there is a certain history. When we look at the date of the first indicated treatment, it needs to be, of course, after diagnosis, after the first diagnosis. So people might obtain drugs for Parkinson that are for other reasons than having Parkinson. So I wanted to confirm that this is the reason they obtained the drug. We wanted to have a history of 365 days of a full year. And we looked at different outcomes. So outcomes are the conditions associated with Parkinson that represent deterioration of the disease. Diagnosis of dementia is one, falls is other, and so on. And we treated as confounders the information about the pre-existing comorbidities, demographics, and so on. And you can see here that we used this same concept of timeline. The results were published last month in a paper in Frontiers in Pharmacology. And the engine that we used was, we were looking at Parkinson patients, and we compared specific two drugs. So we actually, let me start with, we ran our data on Parkinson's patients. We found close to 90,000 such patients at Explorer's Supermart data, and more than 100,000 such patients in our claims data, market scan data. So this was our starting point. These patients who have sufficient history, who have obtained anti-Parkinson drugs and have a diagnosis of Parkinson, and for whom we've seen a sufficient history at the minimum of a year, and so on and so forth, and for whom we've seen that they obtained other drugs, so beyond the Parkinson-related drugs, so other drugs. And we found that there are 259 different drugs, not directly related to Parkinson's, maybe related to Parkinson's complications or to other conditions. And we requested that if a drug is going to be studied in the data, we made two requests, two conditions. One is that there are at least two prescriptions of the drug that are more than 30 days apart. So this is this other drug is definitely used by the person, by the patient, at least according to the records of buying the drugs from the claims data or from the EMR. And we also requested that so one was this, that they have a certain history, and we also requested that at the minimum there are at least 100 patients who obtained the exact same drug. So if the analysis drops below a certain size of the cohort, we are not analyzing the cohort. And you can see here some characteristics of the cohorts that we've looked at. I'm going to move forward and to share with you some of our results. Our approach was following the next procedure. So we were looking at effect measured by the number of events or the prevalence of events. Let's say the event is diagnosis of Parkinson's related dementia. So the prevalence of this event of Parkinson's related dementia within the trial drug, which is the drug at question, compared to the Parkinson's related dementia prevalence in the control cohort. The control cohort is all other patients that are also suffering from Parkinson's dementia, who are using drugs from the same group of drugs that the drug in the trial is. For instance, if this is if these are drugs like here, the psycholeptics drugs that are called NO5 in the anatomical therapeutic chemical classification system, then we compare each and every single drug from this group to the rest, to patients who are obtaining drugs in this group, but not a drug at trial. The example that I will give is the example of what we found. We tested 259 drugs, but we found that zolpidem from this group of NO5 seems to have an effect on the outcome of Parkinson's dementia. So it means that we had a trial drug, zolpidem, and the prevalence of Parkinson dementia diagnosis within the cohort of people who have Parkinson as well as consume zolpidem was high, was in this case lower than the prevalence of the diagnosis of Parkinson dementia within the cohort of people who have Parkinson and who obtain similar drugs, but not zolpidem. So drugs from this same group of NO5, but not zolpidem. And in order to make sure that we are working with the right concept and in the right balanced two groups, so we aimed at calculating the prevalence of a trial and alternative treatment, but of course we had real world data and we had to balance the data in order to be able to extract the empirical difference between the two prevalences. In order to extract this empirical prevalences, what we've done is that we first identified major confounders from the list of hypothesized confounders by their association with the outcome. So we calculated the association between confounders and outcome. And then we computed balancing weights for the treatments and the control cohort. And we tested if the balancing weights are indeed leading to a good balance between the two groups, the treatment and the control. And you can see here how we define the variance that we want to minimize. If it was not well balanced, we did a positivity test. So we looked if there are specific groups of patients within the trial drug who seem not to have an equivalent within the control, and we removed those patients. We actually also applied a systematic way of decision tree in order to identify those specific groups and to eliminate them so that we can balance the data. And only after we convince ourselves that we have balanced data have we tested the difference between the outcomes, and we use this measure in order to find the drugs that seems to have an effect on Parkinson's related outcome. What you see here are two things. First is the Kaplan-Meier at right in the graphs. You see the Kaplan-Meier graphs of the two drugs out of the 259 that were found to seem to have an effect in both data sets, in market scan explorers, in the claims and EMR's data. And you can see that this was razagiline, which is known to help Parkinson for a different condition. And here we've seen that it seems to be effective also for this specific outcome. And the other one was the zolpidem that I just talked about, which is a drug known to be useful for sleeping problems. And there were, in the literature afterwards, we found that we corroborated our analysis with what we found later on, that there was evidence in the literature that zolpidem seemed to have some beneficial effect on Parkinson's patients. But in the papers we've seen, the claim was that the data is not big enough in order to make an assessment and to find that this is significant. What you can see in our study is that we had hundreds of patients in each group. So you can see the actual numbers in market scan explorers listed below. And therefore we were able to show an effect with a p-value that shows that this effect is significant. So with this, I'm going to finish this part, and I'm happy and open for questions. Let me just summarize. The results are available in this paper that was published last month. We have found with this system that accelerates the discovery of drugs that can be repurposed, we have found the two specific drugs using causal inference that seems to be strong candidates for repurposing for Parkinson. Thanks a lot.
Thank you, Mikhail, for this very interesting talk. And now we have time for questions. You can ask them through the question and answer window here. I'll start with the first one that was asked during the talk. Thank you for your very interesting talk. Parkinson is extremely diverse and its subphenotypes, as far as I know, not yet completely understood. Did you account for this heterogeneity in your study?
It's a very good question. And the reason that it's a very good question for me is that we are now dwelling on this question. So we are aware that Parkinson is indeed very diverse. And we do know that we need to be very specific when we look at the phenotype landscape of Parkinson. And that's why when we did our analysis, we selected very specific targets like the Parkinson-related dementia and Parkinson-related falls and so on. We are now analyzing the data from the perspective of the phenotype subgrouping. And I can't really report to this is very initial. I agree this is an important question and needs to be addressed, and that's what we are doing as a follow-up to this study.
Thank you. Is there another question? If not, I have a question. Is there a multiple testing issue in this search for treatments and alternative treatments, Mikhail? Or do you somehow restrict the number of potential drugs that you are looking at, and thereby you limit the number of pairs that might be discovered in such a big data set?
So thanks for the question. This is another area at which we now explore. So what we did in order to limit the number of unnecessary tests, so if you test for something that is not likely to be powerful, you wasted an opportunity, because then we will have to correct for this testing. So we want to eliminate unnecessary tests that will not end up with a stronger hypothesis. So the first elimination, the most simple, straightforward one, was what I mentioned, that if we don't have sufficiently large cohort of patients for this specific test, we are not going to perform it. So having more than 100 patients that meets all the criteria, that is, they all obtain the drug or they are all obtaining other drugs from the same group, so that we have sufficiently large case and sufficiently large control groups, and 100 was our criteria, was how we addressed this effort to not perform unnecessary tests in order not to lose the power. We, of course, in all the results, we corrected for the multiple hypothesis testing. We are now looking into more sophisticated ways to make an estimate upfront on the power that an experiment can provide us with, because we thought that this very rough estimate of the size of the cohort is not reflective to the other elements that plays a role, like the prevalence of the specific outcome that we are of interest. If the prevalence is very rare in those cases, we probably will need more than 100 in order to see the effect. If it's very common, then we might be able to work with even less than 100, or a different, maybe it should be more than 100. We ended up with a lot of tests that were not significant. So we only report on those that were significant, but we did correct for multiple hypothesis testing with a very large number of tests. So it's a good question. We are now looking into it.
And there is this other line of work on drug repurposing, which is using biological networks, so networks of relatedness between drugs and diseases and so on. So have you considered that or thought about that, of like grouping drugs based on their relatedness through one of these biological networks or drug disease relatedness networks?
Yes, so our colleagues from the IBM research in Zurich are looking into a technology called RoboXN. RoboXN is a technology that aims at identifying molecules, new molecules. So from scratch, you look at them from the basic principles of what you want to find within the molecule and what characteristics and functionality you need from the molecule. And they find the 3D structure and its function. So this is, even before you look at the biology, that's the first step. So you might want to look at different molecules from these very first principles. And there are different methods of transformers and generative models in order to come up with this proposed chemical structures. Then the next step is looking into, as you said, the biological interactions and making some simulation or emulation of how the novel molecules will interact with this, to do pathway analysis, interact with proteins, docking stations, and so on. So we are looking into closing the full loop. From basic principles, what molecules seems to be of value. Then from the simulation of the pathway analysis, what probably can address against target, then finding if the same molecules were already found or are part of the system, and closing the loop by performing this real-world evidence-based analysis in the event that the molecules are only in the market. So we are looking into this full close, yeah.
Thank you. There's another question by Damian Rocairo. A more general question: are there ways to estimate the long-term effects of a drug when it is repurposed for a different disease or condition?
Yes. So I want to share that, similar to what the discussion earlier today, we are looking into long-term COVID, and we are asking ourselves similar questions of what can we see in the data that seems to be linked to the acute COVID condition? So the PASC, the post-acute sequela of COVID, is a very important question nowadays across the world. And we want to know if we can tie different conditions and phenotypes in the data to the COVID, and then to analyze potentially harmful as well as beneficial effects of different drugs that people with comorbidities obtain on this long-term condition. So this is, again, for me, it's a very good question, because we are looking into it, and probably the best way to identify long-term effect is the comparison in large scale. So the fact that we have available to us data coming from millions of patients, and unfortunately in the case of COVID, it's also a very large number of patients who have had COVID and now are being tracked and supported by different health services. And the comparison of the statistics, the one comparison we performed already, and we have an initial, a very first paper on that in medRxiv, is just the effect of COVID on tens of thousands of patients from US that we've seen in Exploris. So the effect on COVID that we can analyze based on comparison of baseline data of 2019 and actual data of 2020. So when you get to these very large numbers, the background information, the statistics of the population from a year before is very useful in order to link the novel or the differences in the statistics to this unfortunate new condition that we all suffer from in 2020.
Thank you very much. If there are no further questions, then I thank you for this wonderful talk, Mikhail. I also thank Patrick again for his exciting presentation. This was a great symposium, which I enjoyed very much. And I'm very happy that so many people dialed in and joined us today and joined our discussions. Please watch our schedule for upcoming eSymposia. There will be more exciting symposia from my department in the future. You're most welcome to attend these as well. Thank you to the keynote speakers. Thank you to the audience, those in particular those who ask questions, and have a good evening. Thank you very much. Bye-bye, and this will be available as a video on YouTube later. Take care and bye-bye.